Okay, hi, my name is Ricardo Borges, and this will be walking through DAFdiscovery, how to run it. So this is the interface. It's a Jupyter notebook. They are all, it's all written in Jupyter notebook, so it's really easy for others to follow. So let's go. This is just information for cells information. These information are marked as markdown text, and these little clear yellow maybe background cells, they are marked as code. You can see the code here, and these would be marked down. Okay, so let's go. To run each cell of code, you can run using this button, which will run the cell and select the next cell, or you can just go for Control Enter. This is what I rather to do. So this is the dependence installation and importing. This is something that you have to import outside or even here, but you can import outside. And once it's installed in your computer, you just need to import those Python libraries that you will need. Okay, so let's go more information. This is a cell where you do where you select the project, and you will navigate to the project and then other cells. This is the general workflow. I'm going for specific cases just because I would recommend users to start from the general and then understand which option you can use for data merging and correlation calculations. So let's go for some examples. In case one, this case, we have, let me run that, Control Enter, let's run case one. Let me show you case one. This is the organization that I have inside the DAFdiscovery folder. I have several cases organized as directories inside the main folder. The notebooks are saved outside. The STOCSY function here is here, and it will navigate to each project folder for doing things. So that would be the project folder. The only thing we need is the metadata and the NMR. So the metadata will have, let me show you really quick. We'll have sample IDs and the file name for each technique. In this case, this is just NMR. This is a NMR data that I downloaded from metabolites, I believe, or metabolomics workbench. It's on the paper. Okay, so this is the files that you need. So let's go. You can navigate to the folder. If you do ask B, it will create another cell. You can then, just in case, just to show that we navigated to inside the case one, which was the case that the project that I choose. Okay, we don't need that. So this cell will get the information from metadata, and according to the information that you have in that metadata, you will choose what option you should go. So this is the part where I would, from the general workflow, from this general workflow, I would just select the option and delete every other options, right? To keep it clear, to keep it simple. So in this option, I have just NMR data. Let me run that. That will organize the NMR data, and it will save, it will actually reorder according to the sample IDs that is the list, the sample ID list that is in the metadata. And this part, STOCSY calculation, this is where we do the STOCSY. we have to select a driver. The driver will be one of the peaks, right? And this is a peak that would be identified as epuric acid in this data set. So I decided to select that, and that will show me the resulting STOCSY spectra for that case. And this is the spectra, this highlighted here. This is the spectra of epuric acid. You can check that I selected that peak, and these peaks pop out, are highlighted as being highly correlated to this one and presenting a positive covariance. So this is the statistical pure spectra for epuric acid. We can go for another. If you decide that you need to do more than one STOCSY calculations from more than one driver, I would suggest you to copy the content of one cell, do ask B to another cell, and then you modify the driver, right? Because you want more information. So this other peak would be identified as citrate. So let's check if the highly correlated peak are according to citrate. So that's the highly correlated peak, and that's the profile expected for citrate, right? So this is STOCSY working with only NMR data. Let's run for another example. In this example, I think it's a better. It's a next case. I'm doing Control Enter to run the cells. Control Enter. In this case, we are doing MS and bioactivity. So this would be option 4. Option 4. This is it. Just because in the metadata, we have columns named bioactivity and columns named, and another column named MS file name. So in this other cell, we run using Control Enter again, and that would do that reordering of the columns according to the sample ID in the metadata. And after that, we merge the data from the mass spec with the data from the bioactivity readouts. This is combined into one data set, and this data set is saved as CSV in case users needed to do other things, maybe to apply other type of statistics, right? So the STOCSY calculation will be done. Again, you have to select the driver, but in this case is where you have bioactivity data. You usually want to see which features are correlated with the bioactivity. So you will select always the bioactivity peak, right? And this is the code, the line of code that will select that bioactivity in this case, of course. So I can run that. This figure will not be of use, but we want to produce this file here. This file here will have the MS features added to the correlation values calculated in relation to the bioactivity, right? So in these other cell, we can do this kind of plot. This plot is a scatter plot of retention time versus rho MZ and to Z ratio, and it will show the dispersion of the MS features across the retention time and the M to Z range, and it will color code these circular marks according to the correlation value, right? In this case, we added a line here to make it the size related to the correlation value as well. So you can then rover over the MS features, and you can see which peaks are highly correlated and positively correlated to the bioactivity, and that will enable you to filter your data and to have a more simple analysis when you're looking for bioactivity across samples and using mass spec as a analytical tool. Just run for another example. In this example, let's see what we have. Case 3. Again, inside case 3, we have a metadata that will drive all the decisions, right? And according to that metadata, we have again MS data and bioactivity. We are reordering the mass spec data and the bioactivity according to the metadata sample IDs. We run the STOCSY function using the driver, the bioactivity peak as a driver, and we actually want to see this scatter plot because this is from the features, from the MS features, right? So this MS data is not LCMS, this is from GCMS, and we decide to go for cluster index versus retention time. So now you can see the cluster index just because it's easier for GCMS. It's easier to go for cluster index instead of the M to Z ratio, just because you don't have selection. In this case, this is not an SMS, so you don't have selection for the ionized species. This is done up, just to remind you, this is all the MS data is done after the convolution, right? The right processing, as you would do with MZMine 3. So you can simplify your view again, you focusing on just peaks that are highly correlated with the bioactivity. This simplifies your view and simplifies the way you analyze your data. The idea is to make it quicker, faster, right? Let's go for another example. Case 4. Inside metadata, we have NMR and bioactivity. Reordering data according to the metadata. Run STOCSY from the bioactivity. And now this plot will be important. In this case, we are dealing with a situation where the bioactivity data was measured as minimum inhibitory concentration. So we actually want to see negative covariance and highly correlations, but in that area, negative correlations, right? This is just indicating that the concentration of the peak, the higher some compound will be, the bioactivity compound will be, the lower the MIC will be. So we want to see an indirect correlation. So we can select these peaks here. Those are peaks that are correlated to the bioactivity. In this case, correlated to lower values of MIC. For this study, this was the case. And one last example. Case 5. Now we are merging, this is interesting. Now we are merging NMR and MS. Let's do the reordering of data. Sometimes when you process data, your processing, it puts samples out of order, or when you acquire the data, you put samples in a randomized way. So you want to reorder the data, and we do that according to the metadata, because the metadata has the right order of the sample IDs, right? So the stocks in this case, let's select a driver from the NMR peak, from a NMR peak, and the NMR spectra. This is from carbon spectra, right? So the NMR, this is the NMR counterpart of the STOCSY, of the data set, of the merged data set between MS and NMR. So these bright red are highly correlated NMR peaks to this one. This was the one selected as a driver. And we can go for the MS counterpart, and we can see that these bright red circle marks are the ones that are highly correlated to that NMR peak. That's the idea. Now we have NMR features directly correlated with MS features. And we can do the opposite. We can select a driver from the MS side. So I'm asking, in this situation, I'm asking to the NMR which peaks are correlated to a certain MS feature. And in this case, I select a feature according to the post-processing, according to the convoluted MS features, right? So these would be the highly correlated peaks that characterize that MS feature. So this is all the examples that I have for today, and I hope you understand. Well, people understood everything. If there is any things going wrong or any things to learn more about Jupyter Notebook, you can contact me. I'll put a link to my webpage for you to contact me. And yeah, I hope you make use of it. Thanks.